So good afternoon. Do you have any question regarding the DNA repair mechanism that we discussed in our last class? Do anyone have any question? Because if you have any question, then we shall discuss briefly now, and then we shall start the rest of the part of the lecture on DNA repair mechanism, because last class we discussed the cell cycle as well as principle of DNA repair mechanism, especially the major steps of a single base repairing mechanism. Do anyone have any question?
Yes, sir. Can I ask?
May I jump in for those?
Yes, sir. Sir, you used an terminology like replicon. What does that mean?
Replicon,
Yes, sir.
replica. That means during replication, single copy is replicon.
Sir, the newly generated copy.
Yes.
Okay, sir. Sir, another one. Sir, epigenetics. What's the difference?
Epigenetics, very
Yes,
good
sir.
question.
Yes, sir. Sir.
Yes, epigenetics, I didn't talk with you before.
Yes, sir.
Epigenetics means, genetics means basically, we mean ATGC in the DNA sequence if there is any mutation taken place, or we add any new gene, it is we call genetics. Epigenetics means change not in the base, but at the side chain. For example, if in the sugar, instead of hydroxyl, if methyl group is added, then it is called epigenetics. In the sequence of, for example, sequence of two organisms are same DNA sequence, but they may be epigenetically different. That means in the side chain, a base, then a phosphate and a sugar, in total we called a nucleotide. So in the nucleotide, not at the base level, if changes is taken place in the side chain of a sugar or phosphate group, then it is also affect the gene expression as well as the genetics of the organism. It is called epigenetics, not in the main sequence of the bases, but in the structure of the sugar or phosphate. Clear?
Okay, sir. Sir, another one. Sir, is it some kind of mutation?
Is it some kind of?
Mutation, can we say
It
it?
is not mutation, it is epigenetic difference. Mutation we consider if there is any changes taken place in the DNA sequence.
Okay, sir. Sir, are these sir things inheritable?
Hmm?
Sir, are these inheritable?
Another good question. Yes, whether epigenetics is heritable or not. It can
Yes,
be
sir.
heritable or not heritable. Uh, uh, Both can be happen. Epigenetics, is associated with many diseases also, mostly
Okay.
heritable.
Okay, sir. Thank you,
We
sir.
shall discuss later on the epigenetics a bit in detail, but this is very interesting topic for uh, modern genetics. We can even edit a DNA at epigenetic level by using CRISPR-Cas genome editing. Any more question? Very interesting question, Majavin.
Thank you, sir.
If no question, then I shall enter into our today's lecture. In our earlier lecture, we discussed about base excision repair. If there is an excision or any error taken place at a single base level, how DNA can recognize that damage at the base level and then repair it. So there are, major 5 steps. First step, you can see uh, the DNA glycosylase recognizes the damaged base, and then removes base, leaving deoxyribose sugar. And then a purinic or a pyrimidinic endonuclease cuts phosphodiester backbone, and DNA polymerase replaces missing nucleotide, and DNA ligase seal it. So these are the major 5 steps of base excision repair. And this is very, very interesting and important for your exam also. If failure of DNA repair happens in any cell or organism, what can happen? When DNA repair fails, fewer mutations corrected, increase in number of mutations in the genome. So mutation is increased in the genome naturally. The protein P53 monitors repair of damaged DNA. That means, DNA, in the DNA, there are some, administrative process, repair and maintenance department, even inspector like protein P53 monitors, monitoring system, whether repair of damaged DNA happens or not. If damage too severe, P53 protein promotes programmed cell death. This is very important because if DNA repair somehow fails, then P53 monitor and check it, just like proofreading, and then it promotes programmed cell death or apoptosis. I told you apoptosis means to kill the cell in a programmed fashion and remove from the tissue and excrete it from the tissue. So completely damage the cell. After killing the cell, completely damage it and remove from the tissue. So apoptosis is a programmed cell death. And P53 protein monitors if there is any failure in the repair of any base, then programmed cell death occurs. Which cell has such an error? Mutation in genes encoding DNA repair proteins can be inherited, because if mutation is taken place, then it can be inherited from one generation to another if it is stable. Overall, increase in mutations as errors or damage to DNA no longer repaired efficiently. If mutation is too much in the DNA, then correction of the error is efficiency is poor. So DNA damage, cell cycle abnormalities, hypoxia, you can see if damage taken place, P53 monitor, and in the cell division, that cell does not go for division, because if it is not repaired, first repairing process is done and cell cycle restart. After the repairing, if it is impossible to repair, then apoptosis taken place. And cellular genetic stability means every cell contain almost same DNA sequence, so that it can maintain the Homo sapiens as Homo sapiens, that is species integrity. So nucleotide excision repair can also be taken place. Nucleotide, we discussed base excision repair. Excision repair can be taken place at nucleotide level. endonuclease cuts on either side of the damage uh, up to 20 nucleotides together. For example, if damage is taken place at the nucleotide level, then endonuclease cut it. And you see, here is the how to say nucleotide uh, uh, damage or somehow wrong nucleotides, but it is cutting far distance like a surgery. A surgeon does the job like this way. If they want to remove the tumor, some fresh tissues also cut and remove. So you see the endonuclease cuts on the either side of the damage. You see this side and this side. And excise the oligonucleotide. Oligonucleotide means several nucleotides, stands unowned by helicase. And UBRC has the endonuclease activity, and UBRA and B bind to the damaged site. And nucleotide excision repair can also repair other types of DNA damage. So this type of large-scale surgery can be taken place. Similar to previous one, DNA polymerase, recruit or polymerize complementary nucleotides. For example, T for A, like T, like this, and C for G, and recruit here, polymerize, and then ligase pasted it, and it repairs fully. So essentially in human, people with geroderma pigmentosum have reduced NER and extreme UV sensitivity. Also a NER deficiency in NER means nucleotide excision repair deficiency in human promotes cancer. So this type of big, nucleotide damage, if taken place, it can lead to cancer. Probably a minor pathway for dimer repair in organism with photolysis can be taken place. And here UV, you see, UVRC, UVRA, they are actually recognize the damage, and here UVRD participate in with the helicase to completely excise the oligonucleotides. So this is another important pathway of DNA repair mechanism at the nucleotide level. I think like the previous one, it is also very easy. You only need to know the major steps involved here. So if we want to see show you in the DNA double a double stranded structure, you can easily understand if UV radiation, actually UV radiation can cause damage of the nucleotide level. When we expose to UV radiation or other radiation or carcinogen, many causes can result nucleotide damage. And in that case, you see how it cuts and remove the this damaged part with undamaged several nucleotides. And then first step, UV radiation can make two thymines bind to each other incorrectly. Here, two thymines, they make the dimer. Actually, they should bind with the, how to say, adenine, but they themselves made the dimer. And it is a big problem in the DNA double helical structure. The enzyme exonuclease finds the damage and cut the DNA strand. 12 nucleotides are removed here. It can be up to 20 nucleotides. And then DNA polymerase fills the resulting gap. DNA polymerase polymerize this oligonucleotide, and then in a correct order, they place and complementary sequence in other side we can see. And then DNA ligase seals the DNA strand, and it is now repaired. The injury has been dealt with. That means that the injury is repaired. And if it is fails, then it would be very problematic. And in that case, what could be happen? One is can happen the apoptosis, because P53 can recognize it and direct the apoptosis gene for expression, and then cell can be the cell which have such a damage can be undergo for apoptosis. Or the mutation may remain and inherited. In that case, it may cause a consequences of cancer or other diseases in case of human, or other genetic disorders, or it can remain as silent. Sometimes even 2 or 3 nucleotide damage can remain silent if it is not in a major gene. So types of DNA repair mechanism: reversal of damage, excision repair, mismatch repair, recombination repair, error-prone repair, restriction modification system. There are several types of DNA repair mechanism. We discussed about excision repair mechanism. That means it is if it is at the base level or if it is at the nucleotide level. If pays off to learn about DNA stuff. Paul Modric, he actually worked on DNA stuff, really central to Paul Modric's life. He examined a series of enzymes that affect DNA ligase, DNA polymerase, the restriction enzymes in the E. coli bacteria. All individual enzymes. It was very difficult to discover their role and discover them, how they participate, and all the works actually the Paul Modric, Aziz Shankar, they actually did at the using the bacteria first, and then they use the eukaryotic organism. When he subsequently shifted his attention to the enzyme methylase, he stumbled over another piece of DNA stuff that would come to occupy him for a large part of his scientific career. Actually, they spent whole life to discover all these things, and they opened a new area of science, that is DNA repair mechanism. And before their work, people thought that DNA is DNA is stable, and it is unquestionable that DNA damage can be taken place. So methylase, another enzyme, couples methyl group to DNA. This is very important. Some student initial, at the starting point of the research, my lecture asked about the, epigenetics. That means the methyl group or side chain, if there is change taken place, can be methylase can couple it. The methyl group could function as signpost, helping to particular restriction enzyme to cut the DNA strand at the correct location. However, there was a different signaling function for the methyl group on the DNA. A bacterial virus with several occurrences of mismatching bases in the DNA was constructed. For instance, A could be placed opposite C instead of T. So many types of mistake can be taken place, and they did lots of work to prove it, the fundamental understanding. So please go through, these are the slides, maybe, maybe not so difficult to understand the step of their research they followed. But the major learning point is base excision repair and nucleotide excision repair that I discussed in detail. The DNA replication machinery is not error-free. You must remember these fundamental, information. That means DNA replication machinery is involved in enzyme and other, tools involved in DNA replication, copying making a copy of DNA is not error-free. That means there might be some error. There is always the possibility that an incorrect nucleotide is introduced during synthesis of a new DNA strand. That means not UV light or carcinogens, even in the natural process of DNA replication before cell division, error can be taken place. These types of errors are known as mismatches, and they have the capacity to change the sequence of DNA or to introduce mutation. As a first line of defense against mismatches, replicating DNA polymerases contain a 3 to 5 prime exonuclease activity that allows them to proofread the newly synthesized DNA strand. This is very important statement. That means when DNA is replicated, at that time DNA polymerase enzyme is involved to make polymer. DNA is a polymer, so polymerase enzyme is involved to recruit the complementary sequence to the unzipped DNA, single-stranded DNA, and it contains a 3 to 5 prime exonuclease activity. That is exonuclease activity. Exonuclease, another enzyme, it is a proofreader. Check whether the right nucleotide is binding with the right nucleotide or not. For example, whether A is binding to the T or G binding to the C. Otherwise, it would be wrong and it would be detected and it would be corrected. And if it is not corrected, then mutation would be taken place. The exonuclease activity can correct mistakes during DNA replication by reversing the direction of the polymerase and excising incorrectly introduced nucleotide. That means incorrectly, if some nucleotide is introduced, they excise it and remove it and correct it. And you may ask the question, how fast they do the job? They do the job very fastly and like computer program. Even if proofreading efficiently corrects most mistakes made during DNA synthesis, some non-Watson-Crick base pairs still remain. That means non-Watson-Crick base pair means A bind with T, C with G. This is the standard and in the double helical structure, sequence remain, double strand remain like this way for their balance. But even the proofreading is taken place, some base pairs still remain incorrected. To correct these errors, cells use mismatch repair. Today, we know that all but 1 out of 1,000 errors that occur when the human genome is copied are corrected by mismatch repair. That means mismatch repair is another mechanism. If base excision or nucleotide excision is failed to correct all the errors, then mismatch repair is the second phase of correction. However, in human mismatch repair, we still do not know for sure how the original strand is identified, because still this area is dark gray. Scientist is still doing the research how the original strand is identified by the during the mismatch repair. DNA methylation has other functions in our genome to that of bacteria. So something else must govern which strand gets corrected, and exactly what remains to be clarified. The checking and correcting in the process, there are still knowledge gap to clarify, uh, what must go, which strand gets corrected and exactly what remains to be clarified. Despite extraordinary fidelity of DNA synthesis, errors do present. Such errors can be detected and repaired by the post-replication mismatch repair system. That means after the replication, mismatch repair system can do even some errors if remain, and there is a chance to have the error. Prokaryotes and eukaryotes use a similar mechanism with common structural feature. That means in human eukaryote, or bacteria, tiny bacteria, prokaryote. In both cases, fundamental principle is same. Defects in mismatch repair elevate spontaneous mutation rates 10 to 1,000 times. So it elevates the spontaneous mutation rate 10 to 1,000 times if there is a defect in mismatch repair. Defects in mismatch repair underlie human predisposition to colon and other cancer. Colon cancer is a serious cancer and one of the largest cancers for the cause of death. In USA and other developed countries, after the age of 40, every person check their colon by using colonoscopy, and it is one of the most preventive technique to correct if there is anything happens at the initial stage, and otherwise the cancer can cause colon cancer cause the death of the human. So mismatch repair also processes mispairs that result to heteroduplex DNA formed during genetic recombination, act to exclude homologous recombination. So, so that it can maintain the integrity to the parents. Now, problem is, how do cells know which is the right template strand? In E. coli means Escherichia coli. It is a model bacterium hugely been studied at molecular level. New DNA not methylated right away. Mismatch recognized by mute S, then mute L binds and attracts mute H. These are, mute S, mute L, and mute H. They are endonuclease enzyme that clips the mismatch. Clip means cut the mismatches and nearest CTSE that is not methylated. That means these four base pairs, nearest one which is not methylated. If methylation is taken place, then mute S, mute L, and mute H, they detect endonuclease and cleave it, cut, and remove the methylated part, wrongly methylated. That means epigenetic level changes. Eukaryotes, including Arabidopsis, the model plant, had mute S and mute L homologs, but no mute H in case of plant. Also have the requisite, endonucleases, but not clear how the strand specifically is determined. So this is the mismatch repair. We previously showed you the base excision as well as nucleotide excision. Here you see the methylation taken place in the bases or DNA sequence, and these are wrong thing. So in that case, mismatch base is detected by mute S, mute L, and mute H. In E. coli, A of each GATC or ATGC, whatever you see, is methylated. You can see here is the methylation, here is the methylation. So these endonucleases, they detect and cut. Here you see it is cutting. Then methylase is delayed for about 10 minutes, then it methylates the new replicon. You see, it is cut because it is wrongly, methylation is here, and this methylation is, if it is wrong, then it is cut, and mute L, mute S, mute H, they participates, and then correct it. DNA polymerase and ligase, they sealed it. This is the way, mismatch repair. Mismatch repair can be taken place, and it is now a corrected one. So DNA damage and DNA repair, there should be a balance. DNA repair, if properly taken place, then genetic stability is maintained. But defective or incomplete DNA damage, genetic instability taken place, genetic divergence, for example, in the rice Oryza sativa, we have 8,000 of different genotypes in the gene bank of BRI, Bangladesh Rice Research Institute. They are genetically divergent because not exactly same DNA sequence, and it happens obviously through the DNA damage, and this mutation resulted the stable genotypes with different properties. Some are aromatic, some are non-aromatic, and they are stable, can be stably generate the diversity. But in some cases, cancer or hereditary diseases can be taken place. Even in other organisms, some disease or abnormalities can be taken place, which is lethal to the species. So the diversity we can see in the earth, it is due to the inefficiency of the DNA damage repair by different methods. So disease in which DNA repair that is happened in human: geroderma pigmentation. Patients are hypersensitive to UV light. Patients often develop malignancies of the skin. Malignancies means cancer. Ataxia telangiectasia, patients are sensitive to gamma irradiation. Patients develop neurological and skin lesion. Fanconi's anemia, patients demonstrate aplastic anemia, growth retardation, and congenital anomalies related to the deficiency in repair DNA crosslinks. So these are the disease and DNA repair defects can develop. Here you can see the symptom of geroderma pigmentation. You can see on the skin lots of skin problem when they exposed to sunlight, and early onset of skin cancer can be developed. And here you can see lots of symptoms. So why are geroderma pigmentation patients are sensitive to sunlight? Because healthy DNA, UV DNA damage, and 8 genes, they are involved to repair nucleotide excision repair and become healthy. But in case of geroderma pigmentation, they have no correct repair mechanism. This is why they cannot maintain the repairing system, and as a result, they are highly sensitive to sunlight. Another disease is sickle cell disease. This is a very good illustration of the devastating effects of even tiny damage or changes to the DNA. Red blood cells, hemoglobin has a large protein component in the red blood cell. 2 beta globin chains are present. A single base change substitution causes the disease. Here you see sickle cell disease, single nucleotide. Only a single nucleotide change A change to T, cause this dangerous sickle cell disease, and it is very problematic. And some agents damage DNA directly, chemical and light. Chemicals like alkylating agents, because DNA has the base, base pairs. Methyl and ethyl groups added to DNA bases. It can also cause damage. This type of damage can be repaired by direct reversal involving specialized enzymes. They remove the offending atoms and restore the bases. So just a few types of damage is repaired by a simple reversal of the chemical changes, like UV light induced dimers, methylation of bases, ethylation of the bases. These are the damages. How many types of damages can be taken place? Dimer can be taken place due to UV light. Methylation can be taken place in the nucleotide. Ethylation can be taken place in the nucleotide base. Large chemical group added to the DNA. Most other damage require other systems for the repairing. Deamination, an amino group of cytosine is removed and base becomes uracil. You see, only changing an amino group in the cytosine can result the uracil, and deamination is one of the form of DNA damage. Deamination, an amino group of adenine is removed and the base becomes hypoxanthine. Hypoxanthine can, if only one amino group from the adenine is changed. So deamination is one of the mechanism. An amino group of guanine is removed and then the base becomes hypoxanthine, and it is a damage. So deamination is one of them. Another is depurination. In the purine, the base is simply ripped out of the DNA molecules, leaving a gap like a missing tooth. Then it is also a damage. So lots of damage can be taken place. Basic mechanism is the same for all three types of damages. For example, damage to the stop strand, damage detection, excision, and then repair, and then sealing. So in all cases, first remove the damage, resynthesis of the DNA to place it, and then ligate and pasting it to make the repaired one. So basic principle in case of all the damage repair is similar. So summary: Thomas Lindahl, Paul Modric, and Ajit Shankar have made fundamental and groundbreaking discoveries on the enzymatic mechanism of DNA repair. Lindahl demonstrated that DNA is an inherently unstable molecule subjected to decay even under physiological condition. That means without any carcinogen or UV, even during the replication process. Guided by this observation, Lindahl identified a completely new group of DNA glycosylases and described their role in base excision repair. Modric transformed the field of mismatch repair from the genetic observation to a detailed biochemical understanding, as I described, first in bacteria and later in eukaryotic cells. And Shankar has transformed the field of nucleotide excision repair from genetics and phenomena in cell extracts to a detailed molecular description of the mechanisms involved, first in bacteria and later to the eukaryotes. And Shankar also explained the molecular mechanism underlying photoreactivation and the first form of DNA described. So these three guys, they shared the Nobel Prize in 2015 for their fundamental discovery. And I showed you very fundamental understanding of DNA damage, as well as how the damage can be repaired. But the science is moving very fast, and this is a new area of research. Scientists discovered lots of other, factors, enzymes, other cofactors involved in the details of DNA damage as well as DNA repair mechanism. As you are an undergrad student, I think the lecture I delivered is enough for you to understand this, fundamental mechanism. Thank you very much for listening the talk, and we can now discuss a bit about the your questions and comments. You may ask a question, you can make comment. If you understand very well, you can express. If you do not understand a specific point, you can raise the question, because asking the question is helping yourself and your friends to understand the topic very clearly. So it is your turn now. Unmute yourself and ask the question.
Yes, sir.
Yes, Mahjabin Ferdos.
Yes, sir. Sir, you told about the heteroduplex DNA. What does that mean?
Hetaru,
Heteroduplex DNA. Duplex
heteroduplex
DNA.
duplex DNA. Yes,
Yes.
duplex means duplex house. So DNA can be duplex also. Double helical structure is somehow due to the damage taken place. Hetero means homo and hetero. For example, different base or neighboring base, if they pair together, they cannot pair with the complementary other side of their complementary part, and it can result the heteroduplex DNA, and it can also create a serious problem and difficult to repair. In that case, apoptosis is the final treatment, and if it is not happen properly, then it can create a serious, genetic consequences. Thank you for a good question. Any other? Any more question from other student? Or Mehjabin, you can ask more question also.
Sir, in case of DNA excision repair, you said about UVRC, A, B, C, D. What are those actually? I want to know.
They are actually UV light. UV light, different type of UV light. The scientists use to make the damage. And uh, and depending on those light, specific enzymes are involved in repairing the damages. Any more question? I suggest all of you to visit the nobelprize.org to read the press release which described in a popular form about the discovery of these three scientists. So in a popular language, not the technical one I delivered here, if you go through in the nobelprize.org, you can find in details about their discovery. You can get, because it is prepared for general mass people, not for the biotechnologist. I hope it would be helpful, and it would help you to ask me question even in the next class or through the Facebook group. Any more question?
Sir, I have a question.
Yes, Rojana.
Yes, sir. What's the difference between base excision and DNA mismatch?
Base excision and DNA mismatch. Base excision and DNA mismatch. Base excision taken place if a single base or a nucleotide excision repair. Base excision means if a base is somehow affected or defected, and mismatch means not matched well with the complementary other side of the bases. In that case, different mechanism is involved. Detect the mismatch part, and do the surgery and remove that mismatched part, and then polymerase involved in polymerization of the right nucleotide and place it, and then ligase seal it. So
Sir,
please,
DNA
yes.
mismatch can be the base excision,
Uh, uh,
like
mismatch and base excision. Base excision only at the base level if base is damaged, structurally damaged or like that. But mismatch can be different types. I gave you, the illustration so that you can understand where the mismatch is taking place. Not matched well A, not matched well the, T, but other part remain the same or like that. So mismatching, if happens in area, then it is corrected. And mismatching even can be happen in the epigenetic level also. So in that case, different mechanism, mismatch repair mechanism is involved, different enzyme is involved. So they first detect what kind of damage is taken place, whether is base incorrect base or incorrect nucleotide, or mismatching in the DNA double helical structure. Then different enzymes participates to correct it. So please go through and read the slides. I guess you would be much more clear. And even though if there is a question, we can you can discuss through the Facebook, as well as even you can ask me question even next class. One point is that please visit the Khan Academy. Do you know the Khan Academy? uh, uh, Salman Khan, a Bangladeshi born scientist, he developed a virtual university like. So you can find, I'm sure, a DNA repair mechanism in an animated format, in a detailed way, in a video, step by step. He has a very nice animation, not like I showed you the PowerPoint slide. So you can see how things are happening through animation would be helpful. If someone visit the Khan Academy and if you find it in his virtual website, please share the link through our Facebook group so that other students can get more insight about the mismatch repair. Any more question?
Sir, I have a question outside of the lecture today, and it is on mostly based on my personal inquiry or experience. You know, we know lemon. Lemon is a cross self-pollinating crop actually, but there may some planted a seed lemon, and side by happens between the seedless variety and seeded variety. Is it possible that the seedless lemon variety bears seeded lemon?
You
If
mean seedless variety if we want to convert into the seeded one, whether we can do it or not?
no sir, by accident, if cross pollination happens between the seeded variety and seedless variety, and the flower belongs to seedless variety, but is it possible that seedless variety then bear seeded lemon? Is it possible?
Naturally, naturally not, because seedless and seeded in many crops, commercially we love, for example, guava, even lemon, and many other, vegetables even, we want to see them seedless. When I was a student like you, I had opportunity to work at Bangladesh Agriculture University to develop seedless thistle gourd. That means kakrul. Actually, by using, accidentally it can be taken place. That means it is associated with the, how to say, the diploidy, triploidy, for example, ploidy level. It is not at the only a single gene is not regulating like that way, so that during crossing it can be transferred. It is much more than, for example, by using colchicine and other, for example, a triploid, a diploid, if you can make it triploid, it may not produce the seed. For example, haploid, single, chromosome, if present, or double haploid, you cannot find the seed. So it is at the level of ploidy, not at the level of the single gene. So crossing cannot result like this way. But what you can do, you can do seedless one. Seeded one you can make seedless, but I have no idea whether seedless one you can make a seeded one. It is much more complicated.
Sir,
By
how
using
it
different
can,
mutagenic agent, chemical mutagen, or for example EMS or radiation, gamma ray, you can make change at the ploidy level. But ploidy level change not precisely, randomly can be taken place. If you test several thousand seeds, you may find one or two desired, for example triplet, or how to say, or double haploid, or like that. In that case it cannot form the seed.
yes sir, thank you sir.
Welcome. But it is a very good question. We shall discuss later on at ploidy level, how can we make seeded one seedless, or whether seedless one can be converted to a seeded one or not. This is a very interesting question, but people always do the job that is seeded one, especially food crop, either fruit or others. People want to get the seedless, because seedless guava or seedless, watermelon is very expensive, or seedless orange is very good, lemon. But in case of, seedless lemon, seedless orange, what you can do, you can use the vegetative propagation, and you can maintain the seedless progeny and multiply it very easily. But they cannot, cross-pollinated. They cannot cross-pollinate because as they cannot form the seed, and they are basically infertile.
Thank you sir.
Okay. Any more question? No question? If no question, then I should thank you for listening and asking some very good question. And I added one video lecture that I delivered last year on autophagy. Autophagy means recycling of waste material at the cellular level. And scientist Professor Yoshinori Osumi from Japan, he got the Nobel Prize in medicine. So please listen that lecture, which I uploaded in the YouTube. So before, it is very fascinating. I hope you will enjoy that lecture. And later, in the class, I shall discuss more in depth how recycling of waste materials at the cellular level is taken place so nicely, so that interior of the cell always clean and functional. But if you are going to old and older person, the recycling process is poor, the aged person. And this is why you can see the different symptoms of old person. So in your case, or in our case, if we can activate the recycling system of the waste in the cell, we can keep us younger for longer time. So this is really a fascinating topic I added newly in this course. I hope you will enjoy. Thank you so much.